Hello everyone, welcome back to the class. In our previous class, we had started our discussion on fine structure of gene, which is a new concept. And in this module, we'll be learning about different aspects. So in our previous class, we had started by genes, which was the word coined by Wilhelm Johansen. It is a functional segment of genetic material locatable region of genomic sequence unit of inheritance. It is associated with regulatory regions, transcribed regions, and other functional sequence. So it is a unit of inheritance, and these genes are transcribed into mRNAs, which encodes for proteins on translation. So number of size of genes was also discussed. The portion of genes was discussed, the non-coding, which is known as intron, and coding, which is known as exons. So non-coding do not codes for anything. It is an intervening sequence between the exons, and for further modifications or some changes will happen in that intron excising or intron cutting will be happening. So they are the nucleotide sequence which are represented in primary transcript of gene. After this primary transcript of gene has undergone some modifications, so introns are removed. So that is fine, it is removed, it is not present in the final processed form. And examples of common introns are GUAG introns, AUAC introns, group 1 introns, group 2 introns, pre-tRNA introns, RKL introns. So many genes like interferons, histone genes, ribonuclease genes, heat shock proteins genes, G protein coupled receptors do not have introns. We had discussed about the gene families also. Gene families are the set of genes with sequence homology descended from a common ancestral gene, that is known as gene family. Members of gene families are closely related or even identical. They may be clustered together or dispersed in different chromosomes. Gene families are highly variable in size, sequence, diversity, and arrangement. So based on the diversity and functions of genes within the family, so gene families are classified into multi-gene families and gene superfamilies. In multi-gene families, the group of genes are related with related overlapping functions. Here there is no non-overlapping functions. And under multi-genes there are subtypes like simple multi-gene families, complex multi-gene families, which refers to the families in which all the members have identical or nearly identical sequence, families in which members are similar in sequence but different from the gene products to have distinctive properties. And also we discussed about the different types of genes. The gene is having intron region, which is non-coding DNA sequence, and it is having exon regions, which is coding DNA sequence. And we had learnt about homologous genes. Under homologous genes there are paralogous genes, where genes present in same organism that arose through duplication and divergence. And we also discussed about orthologous genes, where genes are present in different organisms. They arouse through speciation and divergence. And also we came across with pseudogene, false genes. Genes are inactivated because of accumulation of mutations, that is conventional gene. Genes derived from the reverse transcription of an mRNA, and integration of that reversed transcribed DNA is known as processed gene. So homologous genes are genes which share a common evolutionary ancestors. A pair of homologous genes has sequence similarity but do not have identical nucleotides, and also it do not result in a similar or resembling products also. So it is because of two genes have undergone different random changes by mutation. There are two categories already discussed, paralogous and orthologous. Paralogous, same organisms arose through duplication and divergence. Orthologous, genes in different organisms arose through speciation and divergence. So this is a pictorial representation. Pseudogenes is non-functional, false, functionless gene copies derived from the functional genes. So they are the type of evolutionary relic that indicates the changing nature of the genome. Abundance of pseudogenes depends on rates of gene duplication and loss. So it is a common feature in feature of multi-gene families. And there are two classes of pseudogenes: conventional, which is non-processed; processed, which has undergone some changes like reverse transcription of mRNA or integration of reverse transcribed DNA. In conventional, there is no such thing happening. It is inactive because of accumulation of mutation, example globin pseudogenes. And for processed pseudogenes, lack introns and promoter sequence. Acquisition of new genes. So have you ever wondered how the genes are present in your body? So how these genes are acquired in you? So the primary driving force of evolution in all organisms, so it is a primary driving evolution, so it is helping for evolution, it is helping for new characters to emerge from an organisms. So there are majorly four ways of acquisition of new genes, that is by duplication, divergence of the existing genes in the genome, or by acquiring genes from other species, or by gene fusion or fission, or by de novo gene origination. So let us discuss one by one. By duplication and divergence may occur in intact genes or two exons. In intact genes, duplication generates two copies of a gene. Duplication is as simple as that. It generates two copies of genes that are identical, but then usually the copies diverge as each accumulates different mutations. So it is referring to a type of conventional gene where there is accumulation of gene mutation which is causing divergence or which is causing deviation from the original gene. So duplication may be result of how this duplication happens: unequal crossing over between non-sister chromatids of homologous chromosomes, or unequal sister chromatid exchange, or replication slippage. Next type is by acquiring genes from other species. This is referring to which type of gene, that is orthogene, where genes present in different organisms that arose through speciation and divergence. So through vertical gene transfer, it is lateral gene transfer. The gene is transferred from the unrelated genome, not direct inheritance from parents to offsprings. Next one is gene fusion. Gene fusion and fissions. Gene fuse, two or more genes can become a part of a same transcript, that is known as fusion, or undergo fission where single transcript can break into two more separate transcripts. So the last one is de novo gene originations. The new gene can originate de novo from the non-coding regions of DNA. So non-coding regions of DNA in the sense introns. So these are the methods, different type of process through which acquisition of new genes happens. So now what is genome? You have come across this word in previous slides. So what are these genome? Let us define. It is a sum total of all genetic material of an organism. So genome constitute of every information required for your body, required for particular organisms. So it stores biological information. So nature of genome may be DNA or RNA based on the type of organisms. If it is of eukaryote, so the nature of genome will be DNA. If it is viral or other type of organisms, it is RNA. Genome can vary according to eukaryotic or prokaryotic. So there are two types of genome: eukaryotic genome, prokaryotic genome. So eukaryotic genome consists of two distinct parts. So eukaryotic gene, if you imagine the eukaryotic cell, we had discussed about nucleus, we had discussed about organelles. So there are DNA or genetic material which are present in the organelle also, which are present in the nucleus also. We termed the genetic material which are present in the organelles other than nucleus as a extranuclear genetic material. So that is nuclear genome, which consists of linear double-stranded DNA, double-stranded plasmid in lower eukaryotes. So the nuclear genome refers to the genome which are present, the genetic material which are present inside the nucleus. Then is organellar genome, which is extranuclear genome, which is present in mitochondria and chloroplast. So we can say that it is a mitochondrial genome, it is a chloroplast genome. The amount of DNA present in the genome of a species is called C value, which is characteristic of each species. So DNA content of the organism's genome is related to the morphological complexity of lower eukaryotes, but varies extensively among the higher eukaryotes also. So what is this genome complexity? It is the total length of different sequence of DNA. So it can be measured through renaturation kinetics of denatured DNA. So by renaturation, what happens in renaturations? The complementary base pairing again after denaturation of DNA. So denaturation is separating two strands. Renaturation means again complementary base pairing again. So here you can see complementary base pairing between two strands. Denaturation means these two strands are removed or it is separated. The renaturation means after denaturation, DNA is subjected to renaturation process where the base pairing happens again to form this helical structure. Human nuclear genome, organelle genome, E. coli genome, yeast that is Saccharomyces cerevisiae. So these genomes are of different kind. You can come across this information later. So just go through this or glance through this so that you can come across different types of genome, complexity of genome, and special functions of all these genomes. Renaturation and denaturation of DNA. What are this process? So here double-stranded DNA is there. If a solution of double-stranded DNA is slowly heated, the nucleotide strands separate. So this results in the base pairing or breaking of base pairing, complementary base pairing. This process is known as denaturation. The slow heating of DNA causes the two strands to separate, that is denature. If the solution is then cooled, the complementary single strand will come back together, will reanneal, will rejoin, will paste to each other, will attract to each other, will complementary base pairing happens to form the double stranded DNA. So the first process here is denaturation, and the process next is renaturation. Renaturation means the separated strand are sent to or are undergoing again base pairing, again, that is known as renaturation. The rate of renaturation can be followed spectrophotometrically by UV absorbance decrease as duplex DNA is formed. This suggests that we can see or we can monitor the rate of renaturation spectrophotometrically by UV absorbance. So here you can see the denatured getting renatured. So at the end of the reaction, so the COT curve represents the fraction of DNA remaining single stranded in the renaturation. So here the reaction is starting where two strands are separated. By the time when it reaches a particular plot or particular point at the end of the reaction, it will become what? Double stranded at the renaturation. So this curve is known as COT curve or C0 curve. The C0 T curve of a reaction described as a kinetic complexity. It indicates the total length of different sequences that are present. This reveals the sequence in DNA. So this renaturation is very important so that we can elaborately see the DNA, sequences of DNA. Next is reassociation kinetics. So what is this reassociation kinetics? While the complementary base pairing is happening again in between the two separated strands, so at that time we can see the genomic sequence, we can identify two general types of genomic sequence, that is non-repetitive DNA and repetitive DNA. So the genomic sequence will be having non-repetitive DNA and repetitive DNA. How we can observe this? With the help of reassociation kinetics or with the help of renaturation process of DNA. What is this non-repetitive DNA? It is evidently indicating that non-repetitive means it is not repeated. It is a sequence which is unique. It is having only one copy in haploid genomes. They show less rapid renaturation rate. It is present in prokaryotes, lower eukaryotes, etc. Repetitive DNA, it is evidently showing that it is repeated. It is present more than one copy in haploid genome. It shows more rapid renaturation rate. Eukaryotes have half of their DNA occupied by this sequence. So repetitive DNA sequence are more. Under repetitive DNA there are two types: moderately repetitive, highly repetitive. Moderately means half or partial. Highly means more DNA sequence are repeated. Moderately repetitive DNA sequence are relatively consisting of short sequence. They are repeated typically 10 to 1000x in genome. They are dispersed throughout genome. It is significant fraction of moderately repetitive DNA which consists of transposable elements. So example for moderately repetitive DNA sequences, transposable elements. We will come across this transposable elements in our next previous future classes. So highly repetitive DNA sequence, these consist of very short sequence of about less than 1000 base pairs. They are repeated many times randomly in large clusters. So rapid rate of renaturation, a component that renatures most rapidly in eukaryotic genome, they are called simple sequence DNA. Example is satellite. So remember this chart. Genomic sequence is divided into or classified, or the genomic sequence which are present in the DNA is non-repetitive type of DNA, repetitive, under repetitive, moderately repetitive DNA sequence, highly repetitive DNA sequence. So highly repetitive, example satellite; moderately repetitive, example transposable elements. So here the same thing is shown: highly repetitive, moderately repetitive, unique. By the end of the renaturation we can see unique sequence. At the middle of the process of renaturation we can identify moderately repetitive. In denatured condition we can see highly repetitive sequences. Highly repetitive sequences, that is satellite, microsatellites, mini satellites. Let's learn about this. Tandem repetition of short sequence. It creates fraction with distinct base composition from genome average. Difference in base composition creates difference in bioint density. Simple sequence DNA can often be separated from bulk of chromosomal DNA by shear degradation. Cesium chloride density gradient centrifugation separates these DNA on the basis of their difference in bioint density. Formation of satellites to main DNA band. Sequence of these DNA is called as satellite DNA. So what does this refer to? So the tandem repetition of short sequence creates fraction with distinct base composition from the genome average. When this simple sequence, when we take a simple sequence and separated from the bulk chromosomal DNA by shear degradation, with the help of cesium chloride centrifugation, the DNA separates on the basis of the difference in bioint density. With the help of the cesium chloride density gradient centrifugation, there is formation of satellites to main DNA band. These sequences are identified as satellite DNA. What are these satellite DNA? Satellite DNA is essentially synonymous with the simple sequence DNA. Example, when mouse DNA is centrifuged for a density gradient, there is two types of bands may be distinguished. Main band and the satellite. The main band contains 92% of genome and is centered on a bioion density of 1.701 gram per centimeter cube. The smallest peak represents 8% of genome and has a distinct bioion density. So the simple sequence of DNA, when we perform cesium chloride density gradient centrifugation, it separates. So it separates into main band and satellite based on the differences of their bioion density. Further, so this is the satellite DNA. Where can we find this satellite DNA? They are mostly in centromeric and subtelomeric regions in the chromosomes, but also in intercalary portions also. What are the features of satellite DNA? Satellite DNA, that is SAT DNA, is the highly repetitive DNA consisting of short sequence repeated a large number of times. It carries a variable AT rich, adenine thymine rich repeat unit that often forms array up to 100 MB. So satellite DNA forms a different band in cesium chloride density gradient. So here cesium chloride density gradient centrifugation apparatus is there, which forms, after the centrifugation, it forms two bands, that is main band and satellite band. So majority has main band and a smaller part is having satellite band. Satellite DNA, they are highly repetitive DNA. So here you can see GATA consisting of short sequence repeated several times. It carries a variable AT rich repeat unit that often forms array up to 100 MB. The monomer length of the SAT DNA sequence ranges from 150 to 400 BP in the majority of plants and animals. So satellites are present in many eukaryotic genomes, so they may be either heavier or lighter than the main band. So example of satellites are human satellite DNA, alphoid DNA. Repeats are found in the centromeric regions of the chromosomes. It is species specific. A single genome can contain several different types of satellite DNAs, each with the different repeat units. So satellite DNA sequence generally are not transcribed and are located most often in the heterochromatin associated with the centromeric regions of the chromosomes. Next is mini satellite and microsatellite. So this is a subtype under the satellites. So mini satellite forms clusters up to 20 kb in length. So they are partially having 20 to 50 repeat units, each containing about 10 to 100 base pairs. Example, telomeric DNA in humans. What are microsatellites? Microsatellites are the clusters are shorter, which is less than 150 base pairs. So repeat units are 13 base pairs or less. Typical microsatellites consist of 1, 2, 3 or 4 base pair unit repeated 10 to 20 times. So these types of sequences are also called as VNTRs. What is this VNTR? Variable number tandem repeats, due to variation in the number of repeating units at the particular loci. So this is about mini satellite and micro satellite. See, satellite DNA size is 5 to 171, present in the centromere. Alpha satellite size is 171 BP, centromeric region. Beta satellite 68 BP, pericentric region of the chromosome. Mini satellite 9 to 64 BP, at or close to telomere. Microsatellite DNA, that is 2 to 13, dispersed throughout all the chromosomes. In our next class, we will be learning about moderately repetitive sequence, that is transposable elements. Thank you.